Okay, my name is Scott Coyle, and I'm going to be providing the guest lectures this week for the lectures on Ras GTPase signaling. I'm super excited actually to do this. Ras is one of my favorite signaling molecules, and that's partly because it works a little bit different from some of the other signaling systems you've learned about in the class. And so I'm hoping that by the end of this week, you're going to know all about Ras and why we should care about it and why it's so mutated in disease and other things like that. And as a guest lecturer, you can reach out to me through Canvas, or if you need to, you can just uh, send me an email at this address if you have questions about the course or the material or anything like that. Um, and so with that, let's just kind of dive in. I'll give you an outline for what we'll be talking about this week, and then we'll get going. So RAS signaling. So, kind of the goals and the outline for this week's lecture is that in this first lecture today, the Monday lecture, so to speak, the goal is to get you to understand the biochemical basis for how RAS functions as a molecular switch, like you see over here. So, we're going to learn what it means for RAS to be on or off. We're going to learn how the RAS GTPase can be turned on by what are called guanine exchange factors. We're going to learn how RAS can be turned off by what are called GTPase activating proteins. And then we're going to learn how RAS being on or off, how that on state directs RAS signaling to different outputs. And once we have this kind of fundamental understanding of how RAS works from today's lecture, on Wednesday, we'll actually use that knowledge to understand, the logic of how different RAS signaling pathways make use of this on-off type of activity to provide different input-output relationships into the cell. We'll apply that to understand disease mutations in RAS signaling. RAS, as you'll learn, is one of the most commonly mutated uh, proteins, or RAS signaling is one of the most commonly mutated signaling pathways in diseases like cancer as well as other development disorders. And then we'll talk about kind of what the frontiers in RAS signaling biology and research into RAS signaling are. So we'll talk a little bit about drugs that target RAS and why that's been complicated. We'll talk about interesting kind of systems level or synthetic biological tools that people are using to uh, understand these interesting molecules. And then my plan for Friday, I understand that the discussion sections are a little flexible. So my thought is I would pick what I thought was a pretty cool paper from the Ras literature, go through it a little bit, and then you guys can either reach out to me with questions about it, or we can even have a little meetup about Ras in a Zoom call or something like that. So we'll just kind of see where that goes. But so let's get started with today's lecture. So for today, the two main things, or the main thing that I'm going to talk to you about is the biochemical basis by which RAS signals. And by the end of the lecture, my hope is that you'll understand that one, RAS is a molecular switch at the plasma membrane. And when RAS is bound to GTP, this molecule is in an on state. And when it's bound to GDP, it's in an off state. I want you to know that RAS is activated by what are called guanine exchange factors, or GEFs. And I want you to understand that RAS is inactivated by GTPase activating proteins, or GAPs. And then finally, I want you to know that GTP-bound RAS signals by binding to different RAS effectors. And this can elicit different signaling outcomes by localizing components to the plasma membrane, as well as allosterically activating them to change their activity. And at the end of this lecture too, I'll put in a little bonus video that it's optional, but I think it's really interesting, which talks a little bit about the discovery of this interesting signaling molecule. And I think it's particularly interesting because the way in which this molecule signals is a little bit different from kinases and things like that. So understanding how this molecule came to be identified as so important in signaling, I think is a really fascinating story into how science works. So there's a little addendum video attached to the main lecture that you can watch. So let's dive in on this biochemical basis of RAS signaling. So the Ras GTPase is a molecular switch that's located at the plasma membrane. And so at its core, what RAS is a small 21 kilodalton GTPase enzyme. And what that means is that RAS, like any enzyme you might have learned about in a different biochemistry class, binds its substrate, GTP, and once bound, performs a chemical reaction to hydrolyze GTP to GDP plus inorganic phosphate. And because, of course, it's an enzyme, the GDP can be released from the Ras, and this cycle can start over again. And so, at its core, Ras is nothing more than an enzyme. But there are a number of features about Ras as an enzyme that are going to enable it to function as a molecular switch. And one of those is that this Ras protein is located at a specific location in the cell, and that is the plasma membrane, sort of the cell periphery, um, where where external environments are starting to encounter the cell. And Ras is anchored to the plasma membrane, to the cytoplasmic side of the plasma membrane through its C terminus. So this isn't a protein that's embedded in the membrane with a transmembrane helix. Instead, the C terminus of Ras undergoes post-translational modifications that make it much more hydrophobic so that it can associate or insert this C terminal piece into the plasma membrane, anchoring it in this location. And something that's interesting about Ras is that we're going to talk kind of for most of the lectures, we're going to talk about Ras generally, but there are many different isoforms of Ras. For example, HRAS, KRAS, and NRAS. And one of the interesting things about these different isoforms of Ras is that the core GTPase component is highly conserved, but these C termini of the different isoforms are actually rather divergent. And so different Ras isoforms then can end up being differentially modified. And these can do things like change where in the plasma membrane the different isoforms go, as well as potentially modulate the function of these different Ras isoforms in ways that to this day we still don't fully understand exactly how they impact. But we know that these differences do contribute to differences in the function of these different Ras isoforms. Okay, so how do you turn an enzyme that's localized to the plasma membrane into a molecular switch? Like, how do you do that? Well, the first thing to realize about Ras is that Ras is actually going to change its conformation, its structural, the arrangement of the different components of its structure as it moves through the catalytic cycle. So as Ras goes from the empty state to the GTP bound state to the GDP bound state and back, if you look at this structure here, you'll see that there are conformational changes taking place in this enzyme. Okay? And these different conformations can be actually recognized by molecules inside the cell. So there are proteins, for example, RAF, that if you were to say, load up this GTPase with an analog of GTP that can't be hydrolyzed, then and do, say, a pull-down reaction with a protein like RAF kinase, what you would find is that the RAF kinase can only interact with this GTPase when that GTPase is bound to GTP, but not when it's bound to other things like GDP. And so what this means is that there are there are molecules inside the cell that can preferentially interact with the GTP-bound form of Ras, and they do so through what is called a Ras binding domain module. And so if you look at a bunch of different proteins that are involved in Ras signaling, for example, PI3 kinase, RAF kinase, or this RAL guanine dissociation thing, each one of them, in addition to all the various domains that endow these effectors with activity, they contain these Ras binding domains, which are domains that specifically interact with the Ras GTPase only when the GTPase is loaded with GTP and not GDP. And if there are at present over 60 different known effector proteins that can bind to GTP-bound Ras through a Ras binding domain molecule. So this figure's a little bit busy, but what it's trying to show you is if you have this Ras protein loaded with GDP, and they overlay all the different structures of things that can interact with the GTP-bound form of Ras, there's this kind of conserved interface here where these Ras binding domain modules all interact. And so in this way, the Ras GTPase, when activated, provides a platform to assemble a whole bunch of different effector molecules. And so let's start to put this logic together here. What this means is that you've got a protein at the plasma membrane that in the GDP that can undergo a transition from being GDP bound to GTP bound. And when it's GTP bound, this allows for the recruitment of effectors from the cytoplasm to the plasma membrane. And so when this protein gets turned into GTP-bound state, it can shuttle components from the cytoplasm to the plasma membrane. And in this way, it acts as kind of a shuttle or switch to bring components to and from the membrane. I should also add that the binding, in addition to relocalizing components, can also, in some cases, allosterically activate the effectors that it binds as well. Okay, so that's great. We've got a molecule that changes conformation as it moves through its catalytic cycle, and in this GTP-bound state, it can engage these Ras effector complexes. But how do you actually turn that into a regulatable system? One in which you can control when the molecule is in the GTP-bound or GDP-bound state. Well, and so the crux of how Ras is able to do this is that the enzyme itself is incredibly slow. That is, it might take a Ras molecule multiple hours to be able to complete a single round of this catalytic cycle. So while the binding of GTP is fast, it takes a very long time for the molecule to hydrolyze GTP to GDP, and it takes a very long time for the molecule to exchange GDP, empty out its active site to be able to bind a new molecule of GTP. And so what this means is that if we have molecules that could accelerate individual steps in the conversion of GDP to GTP or back from GTP to GDP, then we could specifically control how much GTP-bound Ras there is inside the cell, and now we have a regulatable system. And the molecules that evolution has produced that do this are called guanine exchange factors and GAPs. And so there are molecules called guanine exchange factors whose function is to turn Ras on. And what these molecules will do is facilitate the exchange of GDP-bound Ras, which is the off state, for GTP-bound Ras, which is the on state. This molecule, this GAP, is going to come here and facilitate this exchange. And so we can think about these GAPs as proteins that turn Ras on. In addition, evolution has produced molecules called GAPs, or GTPase activating proteins, whose function is to take the GTP-bound form of Ras and facilitate and accelerate the hydrolysis of this GTP to GDP in the Ras. So these basically have the function of turning Ras signaling off. And thus, if you want to think about Ras signaling in general, then what you have is you have a switch Ras that can go from empty, GTP bound, and GDP bound, where the GTP bound state can interact with these different effectors. And then, in terms of what types of signaling outcomes are produced, the amount of gap that's present in the system is going to control how much GTP bound Ras is getting converted into GDP bound Ras. And the amount of GEF in your system is going to set how much GDP-bound Ras gets converted into GTP-bound Ras. And so the activity of GEFs and GAPs working together with this GTPase can control how much Ras is on and how much Ras is off.